Chapter 1 of a study on contractions of epithelial cell sheets. This video is similar to a research publication. It presents observations and proposes models for a system that has been studied by others. However, references to previous work are not comprehensive, and this report has not been peer-reviewed. Most of the content of the early chapters are available from a preprint server. I declare no conflict of interest since I am not presently employed or associated with any academic or commercial institution. I welcome comments on these results and suggestions for future work. I start this story not with a young colony, but with an older one that is about 10 centimeters in diameter, attached to the surface of a floating dock. The many small black circles that form the rows on the surface are openings to zooids underneath that draw water in and filter out smaller organisms for food. The few large irregular openings are where the water exits the colony. Nearby colonies produce sperm that are carried by the water to fertilize eggs in the interior of this colony. These fertilized eggs are held and nursed to become a tadpole-like tunicate larva. This tadpole has a beating heart and faint parallel cylinders around the middle, which are nascent ampulla. After swimming for about 24 hours, the ampulla move to the front, the tadpole contacts the surface the colony will live on, and the ampulla spread out radially as the tadpole undergoes metamorphosis to a zooid. After about 48 hours, the tadpole has become an adult tunicate, the founder of the colony. The ampulla are the 30-plus oval sacs around the perimeter, connected to the zooid body by a vascular network. The three small buds on the zooid, two on the left and one on the right, are new zooids that will grow to become the next generation. The zooid has a peristaltic heart and a distinct circulatory system connected to, but quite different from, the ampulla net. Colonies are embedded in a strong but flexible and almost transparent gel, the tunic. Here it appears as a faint gray haze, the edge of which can be seen along the right side of the colony. Here we zoom in on two ampulla in a similar colony. White cells are clearly visible in the blood as it flows into and out of the ampulla. The thin vessels are actually cylindrical cavities in the tunic, lined with a single layer of epithelial cells that can barely be seen as faint lines. The larger oval ampulla are also cavities in the tunic and appear to be more solid only because many more red and white cells are bound to their walls. The blood cells moving in and out of the ampulla are small in number compared to those immobilized, which varies greatly among colonies. Since blood flows into and out of ampulla, they must expand and contract, but this occurs over a period of about 70 seconds, too slow to be perceived by humans. The same video speeded up 20-fold. Now expansion and contraction of the ampulla are obvious. The two ampulla in the middle are approximately, but not exactly, in synchrony. Look carefully at the vessels, and you see that they also expand and contract along with the ampulla. Now we study contractions in a quantitative manner. To do this, we first make videos of the ampulla. These videos are then converted to a sequence of individual grayscale images, 30 images for each second. The individual ampulla are given identification numbers and the connecting vessels, shown here in red, are carefully mapped by following blood cell movement. Colonies grow by creation of new ampulla, vessels, and zooids. This colony formed in only 50 hours, and in 1 hour will grow noticeably larger. Here is a brief description of the scanning process. A drawing application is used to create a black silhouette on a transparent background, matching the ampulla to be scanned. This defines horizontal, for this image, lines on the 640 by 480 pixel frame that must be searched to find the beginning and the end of the ampulla. To do this, I created a custom plugin from the open source application ImageJ, distributed by NIH. The solid left and white lines, containing about 75 pixels, are the ampulla edges founded by the application. At these lines, the gray level increased or decreased past the threshold. The white and black dashed lines on either side of these define the positions the searches for the edges started and stopped. The thick red line in this graph is the area of ampulla number 1 as a percent of its mean over a period of 2,000 seconds, a little over 30 minutes. The thin blue line is 26 cycles of a sine wave, matched by eye to the red line. A sine wave is the most simple oscillation and describes the behavior of common devices, such as a pendulum. During some time periods, the red line matches the sine curve well, but in others it is a very poor match. The Fourier power spectrum is a common numerical method used to compare a test curve to a sine wave. It is essentially a correlation of the test curve with a series of sine waves of increasing frequency. A pure sine wave will have a single peak in the power spectrum at its frequency. The red line in this graph is the Fourier power spectrum of ampulla 1. There is a peak at 26 cycles, but it represents only 15% of the total energy. The rest is spread out over many frequencies. The sine wave, in blue, has only one peak at 26 cycles. Next, we look at the 3 cycles above the first black bar as a phase portrait. A phase portrait of a quantity that oscillates with time, in our case the area, is a plot of the time derivative of the area on the vertical axis versus the area itself on the horizontal axis. Time is not explicit in a phase portrait, so successive cycles are loops. A series of cycles with the same size and shape form just one loop. The phase portrait of a long sine wave is a single ellipse, or circle, if the 2 axes are scaled properly. Phase portraits are useful in seeing how oscillations evolve with time. In this plot, 3 cycles of the sine wave produce one blue circle, while the contracting ampulla produces 2 large orange loops and 2 smaller loops representing motions which do not repeat. Now we make a portrait of the 3 cycles above the second black bar. This phase portrait of the ampulla, orange line, is almost a circle. Thus the ampulla oscillates like a sine wave in this segment. Conclusion: sometimes the ampulla oscillates like a sine wave, and other times it does not. We couldn't see that with the Fourier power spectrum, which is only useful for getting an average of many cycles. After looking at the oscillation of 1 or 2 ampulla in detail, let's look now at a large number to see how similar to each other they are. Colors are a good method to code areas, since we will need only approximate values. This plot, with 15 vertical lanes, shows the area of the 15 adjacent ampulla in the preceding colony. Patterns in most of the lanes are clearly not regular sine-like waves, and some are very irregular. Ampulla that are next to each other sometimes have similar patterns. For example, 10, 11, 12, 14, 15, 20, 21. That's the only rule I can see. This plot is a color-coded picture of all pairwise correlations between contractions of the 15 ampulla. Red means highly correlated and blue not. The diagonal is all red, meaning correlations of an ampulla with itself. Only one half of the off-diagonal squares are shown because the correlation of amp I with J is the same as amp J with I. The orange and yellow squares are mostly near the diagonal, where the two ampulla are close to each other. The blue squares of low correlations are pairs that are far apart. Ampule pair neighbors that are highly correlated in oscillations tend to beat exactly together for a while, then quickly fall out of synchrony for a while, then just as quickly go back into synchrony. This can be seen here with perhaps a little imagination. The process of falling in and out of synchrony abruptly becomes apparent when we plot the correlation between the two ampulla during a time window of 60 seconds, which moves through the total 2,000 seconds. The running correlation seen in this plot remains at 1.0 for some time, suddenly decreases, then snaps back to 1.0. So far, I have said nothing about a function for ampulla contractions or any reason for synchronization of these contractions. In other words, what does it do for the tunicate? The external vascular net and ampulla are cell sheets, and all cells need nutrients to generate energy. Here, presumably, glucose to burn in order to regenerate ATP from ADP. This glucose can come from only one place, the stomach and intestine in the zooid body, and it can only be transported by the blood. If contractions among ampulla are synchronized, the maximum amount of blood flows between the ampulla in the body. If completely unsynchronized, a great deal of blood just flows between different ampulla. The blue line in this figure is the mean of the 15 ampulla seen in the color plots. This mean has oscillations with a range of about 7%, less than the range of most of the individual ampulla, but still substantial. The orange line in the figure represents the area of the body of the tunicate, which we see expands and contracts along with the ampulla. It is most likely that these body oscillations are due to exchange of blood between the body and the external network. Thus, every part of the tunicate oscillates with the same frequency. The quantitative meaning of the orange curve is not clear, since the profile as seen in the microscope is complex and the body is mostly water. Chaos is an important mathematical concept in the field of dynamics, where solutions of differential equations are studied. There, a system is chaotic if solutions diverge rapidly from each other after infinitesimal changes in their initial conditions. Such behavior means that it is impossible to know the future of a system unless you know the initial conditions with infinite accuracy, which in real-world situations you do not. For our animal, there are not really initial conditions. Life just goes on. Such a system is then said to be chaotic if its behavior cannot be predicted, which in practice means it is not regular. We have shown the system is not regular, not repeating in any recognizable way. Perhaps interactions between the many ampulla and other parts of the animal are just too complicated to result in simple behavior. In addition, the tunicate colony is growing at a rate comparable to our observing times. At the end of 2,000 seconds, the time span of the first plot, The animal doesn't have the same shape and size as it did at the beginning. In the next chapter, the animal will be altered to create regular contractions, and we will develop a different model for the vascular system. Stay tuned.